Hello everyone. Thanks for joining this training course. This training course will introduce how the Sehorse Analyzer works. Sehorse Analyzer is the only instrument which can measure the living cell metabolism. As we mention about the cell metabolism, we will mention the smallest unit of energy. It is ATP. ATP comes from only two resources. One is mitochondria and the other is glycolysis. Glycolysis is a rapid energy resource like our skeletal muscle cell, which will use glycolysis as their main resource. As we mentioned about mitochondria, we are very familiar with it. It is a power plant of cell. So it produces a lot of energy for cells, like our heart, the beating heart. It keeps works, it needs a very stable energy resource. So it prefers to use mitochondria. So mitochondria is much more important than glycolysis. You can think if the cell power plant get explosion or polluted the cell condition, then there is a lot of disease is related of the mitochondria dysfunction. So you can imagine there are a lot of diseases related about the mitochondrial dysfunction, like neurodegeneration disease, obesity, diabetes, or like cancer, immunology function, aging, and the capacity of hepatobiology to in vitro toxicity. So it is very useful a parameter to know the relationship by mitochondria to see the relationship to the disease you would like to research. But it seems not everyone is doing the research of the cell metabolism. Although we think cell metabolism is very important, but we don't do it. The main reason is it is too complicated. It is for a lot of catabolism pathway and the anabolism pathway. So actually we don't know when or how to start to do it. But we can narrow it to a very simple way. We measure ATP resource. One is mitochondria, and the other is glycolysis. Here is a TCA cycle for mitochondria oxidative phosphorylation, and this pathway is a glycolysis. As long as we use the Sehorse analyzer, we can easily to monitor the whole cell metabolism of the whole cell and the change of these two pathways. But major living cell is not easy because cell have to be alive to have energy. If we fix the cell or lysis the cell, we could not measure the living cell metabolism. And it is the reason why the Sehorse analyzer is the only instrument to measure the whole metabolism. Luckily, the oxidative phosphorylation will consume oxygen around the cell. So if cell consume oxygen more faster or more much oxygen was consumed, you can think, oh, this cell have a better mitochondria, a better efficiency. And then in the other way, glycolysis will produce lactate. Lactate will pump out or diffuse out the cell membrane and produce proton. So if the cell produce a lot of proton in a certain time, then it means it have a higher efficiency of glycolysis. So by this way, we use a fluorescence probe, two fluorescence probe, one to measure the change of the oxygen, and the other to measure the change of the proton. then we can measure the whole metabolism of the cell. Because it is very unique, the unique instrument, then we have a unique parameter. Then we will give it a name. We name it oxygen consumption rate. It means OCA. So if your OCA value is higher, it means you have a higher efficiency of mitochondria. And another is ECA, extracellular rate. If you have a higher ECA, it means you have a higher glycolysis efficiency. With this system, we will need to combine necessary consumables. The purpose is to make sure the whole system works stable, because living cell is very easy to influence by a lot of conditions. The cell type, the medium, content, and the temperature, something like that. So we need to make sure everything is stable except the cell you prepared. So you can see the green part is a sensor cartridge. In TMU, the instrument type is XFE24. So you can see we have a 24 layout. Each tip have two fluorescents in the top. And then the bottom one is for cell culture. We will prepare our cell in the bottom of the cell culture plate. If your cell is an adherent cell, you just culture in the bottom. If your cell is a suspension cell, we have a formal protocol for you to attach your suspension cell in the bottom. As we start to do the measurement, the fluorescence probe will drop down to the bottom to create a certain volume space around the cell environment. Then we will use the fluorescence probe to measure the change of the oxygen level and the pH level. We will measure the change per 14 seconds. Then for 3 minutes. Then we will rise our cartridge and do the mixing by up and down to make the environment around cell to the original condition. So the computer will measure the change of the oxygen, translate to OCA, and then measure the change of the pH to translate to ECA. So actually, we measure the whole environment around the cell, and then we can make sure the condition is stable. Then we can make sure OCA and the ECA signal is correct. And then you can see, because we can reverse the environment condition to the original statement, so we can repeat to do a real-time measurement for long term. So by this design, we can incorporate the auto drug injection. So with the auto drug injection, we can inject anything we want, like something you get interested about, like substrate, inhibitor, inducer, toxin, or testing drug. We will use the air force to pump the own drug in the pot you prepared, get into the medium, and then mix by the probe up and down, so the cell will contact the drug very smoothly. And we can measure the cell metabolism change before and after the drug injection. Also, we offer a serious drug injection protocol like mitosis stress test and the glycolysis stress test for you to make more significant difference between each experiment group. First, let's discuss the major application of the Sehorse analyzer. It is about the cancer. A cancer is very close, very important with the cell metabolism. If you are lab do the research of cancer, you should heard the Weber effect. Weber effect, uh, let's describe it easily. Is from a normal cell, you transfer to prefer glycolysis more and your mitochondrial function since is dysfunction. Then it is a transformation from normal cell to cancer cell. We call it a Weber effect. Then we can show or prove it by the Sehorse analyzer easily or by this experiment design. And we see left figure first. We injected oligomycin pre-treated oligomycin. It is a specific inhibitor of the complex 5 ATP synthase. So as we increase the concentration, you will see OCA decreased. But if a cell want to keep alive or make the cell function work normally, you need to keep the ATP supply is stable. So you can see OCA decrease, then ECA will feedback to try to make ATP supply stable. We prepare the same sample to test traditional ADP-assay kit. You can see the ATP supply almost no change. And the glycolysis feedback just another 20% is enough. Then we can see another experiment. We pretreatment the 2-deoxyglucose. 2-deoxyglucose is an analog of glucose, so we can inhibit the glycolysis by a competitive way to inhibit glycolysis. You can see the red line ECA get decreased, and the blue line OCA increased. It is very much increased. You can see almost a 150% increase, but the ATP supply decreased to just a half. It is very weird because we know mitochondria produce ATP is much more efficient than glycolysis. How come it feedback so much but could not satisfy the lack of the glycolysis? So it definitely be the A509, this lung cancer cell line is very reliant on glycolysis, and the mitochondria should be no function or dysfunction, so even they feedback very much, but could not satisfy the lack of the glycolysis. So we call it is Weber effect. So a lot of lab or cancer researchers prefer to try an easy experiment to prove my cell as it become cancer cell. It transfer from mitochondria to rely on glycolysis. So we can see this dot plot. The x-axis is ECA, y-axis is OCA. You can see in this paper of a cell, they research the hedgehog signal pathway. As this hedgehog signal pathway activated, in vitro and in vivo will find a lot of cancer happen. And so as we activate the hedgehog signal pathway, you can see the cell metabolism change from relying on mitochondria, but mitochondria decrease but increase the glycolysis. It means a very standard type of the Weber effect. And then they try to prove it by another way. So they knock down the receptor of the hedgehog. So scramble is the control. This design uses the auto-injection of the Sehorse analyzer, and we call it is glycolysis stress test. Where is the stress come from? It is confirmed in the beginning we change medium with no glucose. So you can see ECA decreased to almost no function. Then we injected the first drug is the glucose. You can see the control group increase the ECA very fast because the cell will uptake glucose by the glucose receptor. Then glucose glycolysis pathway start from hexokinase, then transfer to pyruvate to lactate and produce proton. Also, you can see the ECAG increase very fast. And then how about if we knock down the receptor of the Hedgehog signal pathway? You can see there is almost no increase, almost no change. And at the same time, we can find in vitro and in vivo assay, the cancer happen rate decreased very obviously. So it seems Hedgehog pathway we activate or inhibit it will affect the cancer happen rate very seriously. And we injected 2DG to close the glycolysis pathway completely. Also, it is a very popular experiment design. We call it glycolysis stress test. And in the beginning of the experiment, we will change the medium to no glucose medium. And it is very important. You have to make sure the starvation time is long enough so cell can consume all the glucose inside the cell. But it could be too long to damage the cell. And you need to keep each experiment, the starvation time have to be the same. So it is very important to notice the detail about it. And the first drug is the glucose. Then if a cell get the glucose, they will increase glycolysis very fast. A higher signal means have a higher efficiency of the glycolysis. And the second drug is oligomycin. It inhibits the ATP supply from mitochondria. It means the cell energy all come from glycolysis, no one from mitochondria. And in this stage, the signal may get increased or no difference or get decreased. It depends on a very different condition. If your cell have a good glycolysis function and a good mitochondrial function, and at this time you close your mitochondria, your good glycolysis function will feedback very obviously. If your cell have a good glycolysis but poor mitochondria, then as you shut down your mitochondria, your glycolysis can still work enough. And even because your mitochondria is too poor, there's no feedback is needed. So the signal will keep the same, no change. If your cell have a very poor glycolysis but a good mitochondria, then you can emerge. If we shut down mitochondria, we suggest poor glycolysis could satisfy the energy demand of the cell, so it will decrease. Then cell go to death. Now, so in the third drug is a 2DG to close the whole system. So it is very important to notice in this stage it is not necessary to be increased. It may not change or decrease, also just let you know about it. So it is a very popular experiment design of glycolysis. But we have to say this, let's show you a very good and interesting example about how they use the glycolysis stress test. In this paper, if you are interested about the detail, you can find the original paper. In this research, they find out the different metabolism about the early metastasis and the late metastasis. Let's say a cell travel in the blood vessel or lymphoid cell is just keep the proliferation, but actually maybe they doesn't reach the goal or the target size, so they are not yet prepared to do the real metastasis. But as they reach the target size, the goal, then they will meet the cancer-associated fibroblast. This cell will tell cancer cell, yes, you reach the target size, you reach the goal, and you can start to do the real invasion. So you can see the cancer-associated fibroblast, we just simplified the CAF. You can see they will secrete a lot of signal to let cancer cell know, oh, it is time to do the real metastasis. Then I will break down my glycogen to make my glucose efficiency even more higher. Then I have enough extra energy to do more than just a proliferation and invasion and the real metastasis. Also, let's see how the CAF condition median increased the glycolysis efficiency. Let's see, here is the experiment condition. We divide to 4 groups. One is control without CAF condition median, and this one with CAF condition median. And we add another condition is CP9149. It is an inhibitor of glycogen phosphorylase. It will make the cell don't have extra glucose to do the glycolysis. You can see who is the only one in the top. It is a CFCN. It means with the cancer-associated fibroblast condition median, it will make cancer cell have more glucose from breakdown glycogen to do more efficiency energy transfer. So you can see this is very obviously in this glycolysis stress test. And let's quantify this region. You can see we make the control in one, and we compare to another 2 group with the inhibitor. It has no difference. But the CFCN is very obvious, almost to 2 times signal, very strong signal difference. Then let's do some very common cell functional assay, like proliferation. So still, CFCN have the highest proliferation rate. How about the invasion cell, the number of the invaded cell? It is still the CFCN. So it is a very strong connection of the cell metabolism, of the cell invasion, and it is means, oh, let's more clarify the difference of the early metastasis and the late metastasis. We just said we measure the proton production, and it means the efficiency of the glycolysis. But we need to face a very important thing, is proton is just not from glycolysis. Although most come from glycolysis, but actually there is a part from the TCA cycle. If your mitochondria efficiency is very high, then you will have more proton production from the TCA cycle. It will make you overestimate your glycolysis efficiency. So how could we make it correct? Then the Angelin offer a median. The median, we know the buffer capacity. With this median, we already know the buffer capacity. We can do a lot of calculation because we know how the glycolysis work, how the mitochondrial respiration work, so we can understand how it works. So we did a lot of work, and we have a white paper to prove it. Also, if you are interested about it, you can go the website of Angelin and download a white paper about this glycolytic rate assay. With this glycolytic rate assay, the unit is no longer ECA, it is a proton influx rate because we can calculate the correct proton influx rate by the median we know the buffer capacity. So in the beginning, we will assay cell with a certain nutrient to measure the basal signal. Then we inject the rotenol and antimycin A. This is a complex 1 and a complex 3 inhibitor of the mitochondria. So you will see here is a compensatory glycolysis because the cell just rely on glycolysis now. Then the other drug is a 2DG to close the whole system of the glycolysis. So after the experiment, we will export the raw data to the reporter. The reporter will do the calculation for us. Then it will based on the Oct signal to minus the mitoc acidification. You will get the real basal glycolysis of different cells. And we use this basal glycolysis to compare to the traditional gold standard, the latte production. We can find the relative rate is very high, so we can very confidently say, oh, it is totally work and could replace the traditional latte production. Traditional latte production is not easy to do. You need to collect condition medium for more than 1 day. But so it means it is very hard to collect sufficient median to show the difference of each other. But with a Sehorse analyzer, you can finish the experiment almost just 1 hour, and you can know the certain difference very quick and very obviously to know the difference. And here is data copied from the white paper. You can see this number means the basal glycolysis. And as we plus to 100, then here it means the mitoc acidification. It means we have 7% signal is from the mitoc acidification. The cell line on the left, it is most cancerous cell. So you can see because they rely on glycolysis, so they have fewer acidification or mitochondrial effect. But if your cell in the right side is more primary cell type, not cancerous cell type, you can find because mitochondrial function is very good, very sufficient, they will also produce a lot of proton effect to effect the real glycolysis. So you can see the mitoc acidification will almost to 50% or almost a half. So in white paper, we will say if you don't use the certain medium to do this certain experiment, like the normal experiment, if your OCA divided by ECA is larger than 4, then we will suggest you to do this glycolytic rate assay to make your glycolysis rate more correct. If you are very care about the glycolysis, then we will suggest you to do it. So when you use the glycolysis stress test and when to use the glycolytic rate assay, let me say it more easily. If you just compare in one cell line with a different condition, because it is the same cell line, it is fine to do the cell mitostress, well, to do the glycolysis stress test because you are comparing the same cell. If you are comparing more than 2 different cell lines, then you may have different mitoc acidification. So I will suggest you to do the glycolytic rate assay. It depends on you have just one cell model or more than 2 cell model, and you need to compare each other. So it is the suggestion to choose your glycolysis drug test. Okay, let's go to mitochondria now. This is a cell mitostress test. It is a gold standard to know your mitochondria thoroughly. In the beginning, you will get a very stable signal of Oct, but now we still not call it is not call this 116 a spatial respiration, not yet. The first drug we will inject oligomycin, it will inhibit the mitochondria ATP synthesis, then you can find your Oct decreased. It means there are a lot of oxygen consumption is inhibited, and this oxygen consumption is for ATP production. It is a very important and useful parameter because in traditional ATP as a kit, you could just know the total ATP production. It is combined with ATP from mitochondria and ATP from glycolysis. You cannot tell how many from mitochondria or how many from glycolysis. But with this design, we can know the ATP production from mitochondria, and it is the only instrument to know it. It is very important. And then the second drug is FCCP. It is an uncoupler. It will bring the proton to the matrix, and it will participate to the oxygen consumption. But you need to know, we need to test the best concentration of FCCP. If the concentration of FCCP is too low, you will not have enough proton to increase your oxygen consumption rate. If your FCP concentration is too high, it will penetrate the membrane structure of the mitochondria. The whole mitochondria oxygen consumption rate will collapse. Only the certain FCP concentration will make your Oct to the maximal respiration, and this signal should always higher than basal. We call it SRC, spare respiratory capacity. It means how many space or your ability to face the change of the challenge or the environment. So SRC is a very important parameter to see how good of your mitochondria. And in the last, we inject the rotenol. It's a complex 1 inhibitor, and the antimycin A, it is an inhibitor of the complex 3. It will shut down mitochondria function completely. So the signal over here, we call it is mitochondrial respiration. Under here is non-mitochondrial respiration. So in the beginning signal, we'll need to minus the signal of here. Then we call it is basal respiration. Basal from mitochondrial respiration. So a spatial respiration, here is ATP production, then it should be no ATP production. But why we have no ATP production? It means your proton not come from the ATP synthesis. It is come from the leak of the cell, the mitochondria inner membrane. So we call this proton leak. If your proton leak increased, it means your mitochondria structural integrity is decreased, and it will also affect your ATP production ability because your proton will not come back from here. So it is very important to know the mitochondria. You will get the beginning basal respiration, how many percent produce ATP, and how about your structural integrity and your maximal respiration, the ability to force the challenge, and the non-vital respiration. It is a very good experiment design to let you know your mitochondria. So it is the most popular experiment design in Sehorse analyzer because mitochondria is so important and this gold standard is so useful to let us know the mitochondria. And let's show you a very interesting paper to let you know how could we do to the mitochondria. Let's show the major role of this paper is affected T cell. Affected T cell is trying to kill a tumor cell, but the tumor cell have already produced or built a tumor microenvironment to fight the immune cell. You can see this cell get inside the tumor microenvironment and get a very bad influence. You can see mitochondrial function was decreased, so they don't have enough energy to do a normal function. So the metabolic exhaustion, also this affected T cell could not work, could not kill the tumor cell. So if this tumor microenvironment is so tough to live or even to work normally, so how about we increase the expression of the PGC1 alpha? PGC1 alpha is the upstream of the gene of mitochondria. So as we overexpression PGC1 alpha, we will have a super mitochondria of the T cell. So with the mitochondria of the T cell, we can live well inside the tumor microenvironment and work normally. So we can release cytokine normally, we can proliferation normally, then we can kill the tumor cell. So let's try to prove it by the Sehorse analyzer. In the beginning, we can do a very popular experiment. We use the electron microscope to show you the morphology change of the mitochondria. Here is a T cell from the lymphoid. Here is a T cell from the tail. It means that T cell tumor infiltrating lymphocyte. The lymphocyte we get it from the tumor. So we call it T. T is for tumor. So you can see as a normal mitochondria, it will have a long shape, a very good fusion condition, a lot of crystals, a very long shape. Mitochondria is keep moving system. It will keep doing the mitochondrial fusion and the mitochondrial fission. If you want to have more high efficiency, it will go to the fusion stage. If the mitochondria get damaged, they will try to do the fission. As the fission stage, they will break down the damaged part. They will recycle the part still useful. So you can see if mitochondria is a fusion site, it is a good mitochondria. And here, a mitochondria is fission to a very small part. So it is very obvious. This mitochondria function sucks. So let's try to do a Sehorse analyzer. So we have 4 different conditions. Here is a T cell, the gray circle. And here is a cell line for the positive control, so we can ignore it. And the other 2 is from the lymphoid node. Just another different type way to take it down. So it is very similar. It's just take it from the lymphoid very smoothly, or use a physical force to drain out the lymphoid. So it is almost the same. So you can see in the beginning with the oligomycin injection, but most importantly, to the maximum separation, T could not get good increased. So let's try to quantify here. You can see SRC is the highest signal to minus the basal, from here to minus here. So we get the low is the SRC of T. There's no difference of the lymphoid node. And then you can see how about the ECA. With the good mitochondria, the ECA of this lymphocyte is very low. But in T cell, because the mitochondria function not work, so it have a higher glycolysis. And then let's ignore the T cell from the cell line in vitro. It is very efficiency, both mitochondria and the glycolysis. So in the beginning, we know till the lymphocyte from the tumor have a very serious damage of the mitochondria, very serious damage, and then transfer the phenotype to glycolysis. Then there's overexpression, the PD-1 alpha. You can see we have a higher basal OCA, have a higher SRC, but no change of ECA. So we produce a super mitochondria affected T cell, and we try to cure the mouse with tumor. You can see the tumor area increased slowly, more slower. And the survival rate is prolonged more longer. So you can see it is a very important hint and a clue to let us know how important of mitochondria in immune cell. And actually, in immunotherapy strategy now, there are a lot of people try to produce CAR-T with good mitochondria to make the cure efficiency better. And if we want to cure tumor, we need to know the prefer the, the, the nutrient they prefer to eat. And here is a very interesting cancer cell, HCC. HCC, as we mentioned about tumor cell, we usually emerge, because it is a tumor cell. So they live in a hypoxia condition. They should prefer on glycolysis, and the mitochondrial function should not be very good. Actually, it is very correct in most tumor, but actually, each tumor metabolism phenotype is different. So we should to clarify it individually to make a very precise medical treatment. So let's see, in this HCC tumor, we found ACC alpha expression is very high. The ACC alpha is acetyl-CoA carboxylase alpha. It is a protein. The function is to produce fatty acid, to synthesis fatty acid by itself. So since the tumor cell can synthesize fatty acid for itself to work, then this tumor cell have very high possibility to do the fatty acid oxidation of mitochondria. So you can see, let's try to do two experiments. One is to knock down the expression of ACC alpha, and the other is overexpression of ACC alpha. And then we do the glycolysis stress test and the mitochondrial stress test at the same time. And let's compare the phenotype change of these two different experiments. And you can see the control group is a brown one. As we knock down the ACC alpha, both on glycolysis and the mitochondrial function, the whole cell metabolism decreased. So it means ACC alpha is very important for this HCC cancer. So how about we overexpression the ACC alpha? You can find the control is the blue line. Then as we overexpression, the glycolysis efficiency increased. The mitochondrial function efficiency increased. So ACC alpha is so important for HCC cancer cell. So let's try to cure it. Here is the vehicle control, and here is a treatment of aromatic kinase inhibitor. It is a very common way to treat tumor cell in clinical. Then, because we know they will produce fatty acid and to use it by itself, so let's treat the aristate. Aristate is a fatty acid synthesis inhibitor. So you can find if we do the treatment with a cocktail way, you can find the size of tumor gets shrink very obviously. So it is very important information to let you know. If we know the prefer of the nutrient the cancer cell and the tumor cell prefer to eat, then we can try to do the cocktail treatment of the cell metabolism. So it will have a very good result to show the cure. Then the latest and the newest experiment design I will like to introduce you is a real-time ATP assay. By this assay, it is also based on a median from enzyme. We already know the buffer capacity. As we mentioned before, with the OCA, the blue line, as we inject the oligomycin, we will know how many ATP comes from mitochondria. So we will know the mito ATP, and we can do the real basal glycolysis measurement by minus the acidification of the mitochondria. So we can also get the glyco ATP information. We even can do the acute injection before oligomycin and the rotenone Then we can know the immediate change of the mito ATP and the glyco ATP. It is also depend on a very complicated calculation. So we don't need to think it's so complicated. We just need to do this essay and export the data to the report generator engineering Alfred. If you would like to know more, we also can offer you a white paper. By this way, you can know more than traditional real-time than traditional ATPSA kit. In this real-time ATPSA kit, you will know total ATP. It is the traditional ATPSA kit did. You can know the mito ATP and the glyco ATP, and the unit is the absolute value unit, picomole per minute. By this way, you can classify the cell line in your lab. You can find the cell with higher mito ATP rate, higher glyco ATP rate, or both is very low rate, or both is very high rate. So if you want to do a certain experiment, and you can pick the cell suit for it. And here is another good example. We make here is a metabolome index. Here is mito ATP, divide glyco ATP. We try to figure out the breast cancer cell. One is expression estrogen receptor, and the other is not express the estrogen receptor. So we find out if a cell has an estrogen receptor, it will very rely on mito ATP, but not glyco ATP. So it is a very useful information to let us know the metabolic specific type. And how about the doubling time? If your doubling time is shorter, it means you prefer it sooner. So we can find out the doubling time is not relative to the glyco ATP production rate, but it's highly relative to the mito ATP production rate. It means it should be in most cells if you have a higher mito ATP production rate, then your proliferation rate will get increased. because your double in time decreased. And it is also relative to the total ATP production rate, but it is most rely on the mito ATP, but not glycol ATP. So it is a very useful and interesting kit, and it will let you know more than usual traditional ATPase kit. So if you don't know which kit to pick, here is an easy figure to show you. If you want to know the whole cell metabolism, it is a very good way to start from a real-time ATPase kit. If you focus more on mitochondria, then you have to do the cell mitostress test. And you want to do the further, you want to know which substrate mitochondria to do the utilize. Then you can try the substrate oxidation stress test. Then we will combine the inhibitor of the pyruvate or the glutamine or the long-chain fatty acid. And then if you want to find out uh, the fatty acid more, then you may try to add extra palmitate substrate to the cell. And if you want to see the mitochondria function of each complex, you can try the PMP. It is a pulverizer. It will penetrate the cell membrane, but not the mitochondria membrane. So you can add the electron supplier of each complex, because these are very big molecular. Then you can do a very good experiment design of isolated mitochondria. And if you are care about more about glycolysis, you can do the glycolysis stress test, the glycolytic rate assay. And if your cell is T cell, then we will suggest you to do the T cell activation assay. So it is a suggestion way we will introduce you. And there's another extra application. What we just mentioned is just for living cell. But actually, if your lab is doing a lot of animal model, actually we can do the living tissue, uh, the cell metabolism of the living tissue. But we will need to use another special consumable. We will call it islet capture microplate. In this islet capture microplate, we have a deeper button. You divide your tissue to a small piece and screw to the button, then cover with a mesh to trap the tumor under the mesh in the button. Then we lower down the cartridge to use a fluorescence sensor to measure the OCA and the ECA. But you need to notice we could not to do the drug injection, because the structure of the tissue is too compactness, so the drug cannot diffuse very well. So actually we just measure the basal OCA and the basal ECA, but it is already enough, because we need to do the stress test. It is because cell line, the difference of cell line is very less, but actually the difference of each different tissue of the different mouse, it is very obviously. So it is very good to do just the basal OCA and the basal ECA. Let's show you this paper. This paper is from a researcher in Taiwan. They do a research of calorie restriction. They use a young mouse and a middle-aged mouse, and they try a normal eating and restriction eating. And we show the data with OCA divided ECA. And you can find out it doesn't matter you eat normally or the restriction, you will find the cell metabolism is almost the same. But if you keep to eat normally in middle age, your OCA divided ECA will decrease. but you can keep the same as you do the restriction calorie take. So you can find it the signal decrease here means your cell metabolism change from more rely on glycolysis but not mitochondria. And we move this parameter to the x-axis here, and the y-axis is muscle weight. And you can see with middle age and normal eating, your muscle weight will get down. You will lose weight. You will lose the muscle weight, and your cell metabolism will change from mitochondria to glycolysis. Also, remember to change your eating habits as you get aged. Okay, actually, there are still a lot of application because we measure the living cell metabolism. If you are doing the living cell, you feed cell, you do a cell culture, even you do the animal model experiment. Actually, you can use this Sehorse analyzer to do anything you are currently is doing. So we will suggest you to thinking if this Sehorse analyzer can do help to you. In this instrument, it's very unique to do the whole cell metabolism measurement. We see mitochondria and glycolysis at the same time. It is very important to see these two parameters at the same time because they produce ATP together. If mitochondria change, it will also affect ECA, vice versa. So make sure to measure it at the same time, not individually, not separately. Okay, and then we measure it real time. It means we can do the auto drug injection. So we will have a lot of drug test design to for you, and we can divide to do the deeper side to know more. And it is a proper throughput for 24 well. So you can compare 6 to 7 group with 3 well repeat, and it is very enough. So in this introduction of the Sehorse analyzer, we hope you know more about this instrument, and we uh, suggest you to try it, and it should give you more useful information, and we hope you have the same feeling as Robert. Since you got the device, it should be running non-stop, and it will produce a lot of data for you. So let's take a break. Then following is guide to show you if you want to start a Sehorse analyzer, which consumable you need to prepare, and how to prepare your cell for good, and the detail you need to notice. So let's take a break. Thank you.